Welcome, everyone, to the LACNETS February meeting. We're here at Cedars-Sinai, and thank you all so much. This is wonderful to see such an amazing turnout. I'm Giovanna Mbezi, and we're going on, I think, 3.5 years now for LACNETS, which is a support group for neuroendocrine cancer. And I'd like to welcome everyone, and we have a very special presenter today, Dr. Andrew Hendifar, on a very, very popular topic these days, PRRT, and we're going to learn all about that. I just have a couple of announcements before we introduce him. We've been talking with some other doctors about the programs that they're running in Los Angeles. And two things, City of Hope is going to be doing a full day conference that's for doctors, but they have invited us as well. So our April meeting will be April 9th, which is a Saturday. And it'll be a full day meeting that everyone's invited to at City of Hope. And I'll be sending out all the details. If you're not already on the mailing list, just send me an email at info@lacnets.org, and we'll make sure that you get the announcement. And Dr. Hendifar has been also working with Ken Herman, who's a nuclear medicine specialist at UCLA, and they've invited us next month to meet at their facility. So we will be at UCLA next month. Directions provided, and I'll also be sending out information on that. That will keep our Tuesday, March 8th at 11 a.m., 11 to 1. And then we'll be back at Cedars in May, which will be the second Tuesday. And then in June, we're just working out details, but I think the date is confirmed. We're going to have a very large conference here, as we did last year, led by Cedars-Sinai on June 25th. That's a Saturday as well. So that will be a full day event, and mark your calendars. Is going to be doctors that you will definitely want to see, as well as some guest speakers here. And I think that's it for our announcements. So we will now welcome, most of you know Dr. Andrew Hendifar. He is the oncologist here in the NETTER Center, and so please welcome Dr. Hendifar.
Good afternoon, everyone. Thank you for your patience this afternoon. It wasn't easy to get this started. Appreciate it. Hopefully everyone had some lunch. Today I'm going to discuss some very important results to you. This is from the NETTER-1 study. Some of the patients in this room, no, none of the patients in this room I don't believe have study. But we have had a lot of patients that benefited from this treatment. So we're really fortunate and excited to be able to present to you this today. This has been recently discussed, been really well received. So this is LU dotatate significantly improves progression-free survival in patients with mid-gut neuroendocrine tumors and results of the phase 3 NETTER-1 trial. And LU stands for lutetium. So the NETTER-1 investigator was an international study, which is always important because you don't want to have that can introduce bias. And you see we have 100,000 cases that are active in the United States. That's almost double gastric cancer. So this is a prevalent disease. It's something that we have to do to take care of, something we have to pay attention to. Now, what's really interesting about these types of tumors are that there are several targets, we call it targeted, you can put brackets around it, that we use to treat it, or you can call it hormone therapy. And we all know that we use somatostatin quite regularly. Well, here are all these somatostatin receptors, and we know that many of the neuroendocrine tumors have the somatostatin receptor. And what's an easy test we can use to find the somatostatin receptor? Chromogram helps. What's a test you can order? It lights up.
Octreotide scan.
Exactly, an octreotide scan. So we're very fortunate in this disease to be able to have a test that can tell you the biology, right? There's not that many tests that can do that in cancer. Cancers usually can tell you what a tumor size is, but not many tests can tell you, hey, you've got these receptors on your tumor cell. Okay, so that's an important concept. Another important concept, and yay for us, is that there are a lot of targets for neuroendocrine tumors. There are lots of things that we can do to improve the outcome of this disease. We can target somatostatin receptors, we can target the mTOR pathway, and look up there, I can't reach it, sorry, but the VEGF receptor is up there too. There are lots of things that we can do. Now, to get us excited about this treatment, this is what PRRT, this is a very, not every case is like this, but this is one of the cases that are especially good. Here you see the gallium scan with certain areas in black lighting up, and you have PRRT administered to a patient with a neuroendocrine tumor, and you see before, 3 months after, you see an excellent response with the amount of black areas becoming greatly diminished. Now these two black areas are the kidneys, and this is the bladder, so those don't count, okay? And you'll see here, kidneys, bladder, they don't count. Here's another administration of PRRT, number 2, and again, you see improvement once again. Any questions about that? Does that make any sense to anybody? It's hard to see them for the first time until you get used to these types of pictures. Now we have patients in the clinic too, Most treatments for neuroendocrine tumors, as you know better than anybody, they tend not to shrink tumors as much as keep them stable. And usually that's all we need is to keep them stable. Once in a while though, you'll want the tumor shrinked, especially if it's causing pain. Or sometimes if they're producing the carcinoid syndrome. Then you really want them to shrink down because that'll improve how patients feel. And unfortunately with this type of treatment, you can get response rates, you can get tumor shrinkage, you can get improvement in the carcinoid syndrome, you can get improvement in pain. And that's what makes it so attractive. Now this is not necessarily a new treatment that we're talking about today, but we finally harnessed our resources to get the study done. But this treatment has been extensively studied, and here you see these are different institutions and different studies that have already done it. Probably the most famous is at Rotterdam. They've done lots of research. They've studied 310 patients. That's what N means, number of patients. The average progression-free survival is 33 months, and overall survival is 46 months. Now, what does that mean to us? That's good. I can tell you that's good. That is good. There's no doubt about it. But for patients, it's hard to kind of get a grasp of what that means. But I'll be able to show you later on in this study when we compare it to somebody else. Because sometimes, if you take certain patient populations, those numbers don't mean much in isolation. So you always have to, and that's why everyone hates randomized studies, but why randomized studies are so important, because it gives us the real information that we need to make decisions. So please, if anyone has a question, just raise your hand and just stop me, because otherwise I'm afraid I don't want to lose anybody or anything.
Can you define gut level? You're saying gut level. What is that called?
Oh, midgut. That's a great
Midgut.
question.
What is that?
So midgut is basically the small intestine. Yeah. So there's foregut, midgut, and hindgut. So the way that we decide that is based on the embryology, like when we're fetuses and we separate it out. It's kind of strange. What it really means, foregut is lung, is pancreatic, is stomach, midgut is small intestine, and hindgut is like colon and rectum. Liver is not usually a primary neuroendocrine tumor. They all spread to the liver. It can be. It can be rare. It's rarely. Yeah. But liver is from a different embryology.
So this would apply to what we're saying.
Usually, if you have neuroendocrine tumors in the liver, they either came from the pancreas or the midgut.
Are they usually always identified or sometimes not? Primary?
There is a group that's not identified, but you can still make that assumption that they came from one of the two places.
Pancreas or the.
Mm-hmm. Usually, if you don't know, those are the two sites. Yeah. We could talk about that probably more after. Yeah, you're welcome. Okay, so this is the study design. Here you see 115 patients. Okay, so let's talk about the aim. That's a great. Let's start with the aim. Okay. To evaluate the efficacy and safety of lutetium dotatate and serotonin receptor antagonists compared to octreotide LAR in patients with inoperable somatostatin receptor positive midgut and progressive neuroendocrine tumors. So in order to be eligible for the study, you had to have a midgut tumor, you had to be on octreotide LAR, and your tumors had to have grown by RECIST. Sandra was always working on this part, right? Because the investigators are always like, they qualify, and Sandra had to do all the work and make sure that they really did, right? And the design is an international multi-center randomized controlled group. So these are important definitions, okay? The meaning of the patients have grown, it means that these are not, some patients, you know, the tumors don't really grow on samizdat. But this group of patients, their tumors did grow on samizdat. And was it over 1 year progression or 6 months, son?
Actually, 3 years.
3 years, over 3 years period. So 115 patients were placed on the treatment, and 115 patients were placed on samizdat LAR at 60 milligrams. So it wasn't a regular treatment, it was double the dose of traditional treatment. Okay, does anyone have a question about that design?
Does that mean if you're on laminate you would not be part of the
Right. Which was what they had to do to make it equal across the board. Okay? So again, it was randomized between getting lutetium dotatate, 4 administrations, a 7.4 gigabecquerel, 8 weeks apart, over 32 weeks versus the high dose LAR treatment. Okay, the primary objective was to compare progression-free survival between the two groups. Secondary objectives was to look at the response rate, overall survival, time to progression, safety, and quality of life. Any questions about those things? Yeah, they don't mean a lot to us, right? But these are typically what we do for these studies. So patients have to be greater than 18 years of age. They had to have a neuroendocrine tumor that was growing on standard treatment. They had to have a Ki-67 less than 20%, which is grade 1 or grade 2. So grade 3 tumors were not qualified for the study. Okay? And they had to be somatostatin receptor positive. And remember what we said, that was based on an octreotide scan, or a gallium scan can give you the same information. Okay, so the way these studies are designed are to make sure they're randomized, and it's done by a computer. So the computer makes sure that the two sides are equal. Right? It has to be a computer because otherwise, you know, we'd start taking bribes, etc. God forbid. So the computer makes sure everything is equal. And if you look across the two arms, everything looks pretty equal. The age is pretty good. A little bit of difference in sex. I don't think that matters. The weight is pretty similar. The site of disease is pretty similar. These are important things. Site of metastases. I would say these two, it's very important that they be equal in the two arms. And if you look at the percentage, they're very close. So that means that that's good. The randomization did what it was supposed to do. Look at grade 1, grade 2. So 2/3 of the patients were grade 1, 1/3 were grade 2. Okay? And then this is the Krenning scale. This is how we tell how good the octreotide scan was. How much did it light up? The higher the grade, the more it lights up. And then here's the mean chromogranin and the mean 5-HIAA, which was done as a 24-hour urine collection. Any questions about those things?
How do the doctors and patients not know which one's getting the lutetium?
They did know. Everybody knew.
Yeah, they have to know.
They knew.
It's different treatment.
Exactly.
But the computer figures out, sorts them out fairly, I guess.
So the computer sorts them out before they start treatment. Right. But you're right, they knew in this one. Yeah.
Is the tumor graded 1, 2, 3 according to the size?
No, the grade is based on Ki-67 and mitotic figures.
I don't know what that means.
Right. So Ki-67 is a proliferative index. So what Ki-67 is you take a tumor, you put it under, you take a slice of it like a piece of bread, you put it on a slide, and then you look at how many of the nuclei are turning over, how many are reproducing. It's called a proliferative index.
So how active it is.
Exactly.
Right.
So the higher the percent, 100% means all the tumors are reproducing, 0% means none of the tumors are reproducing. So 0 is low activity, 100% is high activity. So grade 1 and then grade 2 are less than 20%. Grade 1 is like less than 3%. And then grade 3 is between 20 and 100%.
Thank you.
It's very unique for neuroendocrine tumors. Other tumors are graded very differently.
Is that determined only initially when you first have the tumor, the pathology, as you keep tumor slices, that you look at that?
Yeah, usually it's archived, meaning whatever you've had tissue done before. Does that make sense? So if you've had a surgery, even if it was 5 years ago, that's usually what we look at.
That's the case.
Or you've had a resection or a biopsy, but we don't usually do it over again.
Yeah.
Because the way we think is that it doesn't really change that much over time. Although it could, I don't think it's important enough to biopsy again to determine that. Okay. So this is a really good slide. So this is drug exposure. If you look, about 80% of the patients got 800 millicuries. These are words that I'm even new to, because we don't do, I'm a medical oncologist, I do medical treatments usually. So with this study, it's very interesting for me too. Now 79% is an amazing number. Most treatments, most clinical studies I do in cancer medicine, I would be lucky if 30 to 40% of the patients get all the treatment that we had planned for. I mean, the fact that 80% got 800 is really outstanding. It really is. And the number of administrations, 80% of the patients got to 4 treatments, which is the whole treatment, which is really, really great. And this is dose modifying toxicity. Only 6 patients had a dose reduction. I would say on an average study, about 50% of the patients get a dose reduction. So you can compare it to the side effects of the traditional therapies that we use. It's pretty good, pretty favorable. Now this is what we call the money slide. Because this is like the one that everyone's like, oh my God, when they look at this. Okay? So this is what's called a progression-free survival Kaplan-Meier curve. All that means is that on the green line on top are patients who receive the PRRT. The blue line on the bottom are patients who get standard treatment. Now what happens is as patients' tumors grow, this thing comes down, this blue line comes down. Okay? Once all the patients in the blue treatment group, the tumors have grown, the curve hits the bottom. So as you see here, the amount of time it took for patients on octreotide to progress was 8 months. Because that's here. That's the amount of time 50% of patients grew. You come down here, it's about 8.4 months. But the lutetium arm, as of the time of the reporting, we didn't even have 50% of the patients progress yet. And you can see it's already 28 months.
Calculation would be 40 months. 40 months.
Based on now, yeah, they're estimating 40 months. So that's, let me do my math real quick, more than 3 times, right? Now for a cancer treatment, this is really impressive. And their hazard ratio of 0.2. That means that patients, it's hard for me to explain in layman's terms, but let's put it this way, that's a really good hazard ratio. For most treatments to get approved, the hazard ratio is 0.8. So you can say it's 4 times better, the hazard ratio. I mean, it's a very impressive, and the curves being this far separate, usually we say if you can put your hand in between, it's great, and that one you can stand in between the two. I could at least. So I think it's really remarkable. Okay, now the response rate. So we did have 20% of the patients who were on lutetium dotatate had a response, and 3% on patients on octreotide had a response. So it's a big difference in response rate. Please, if you have any questions, please let me know. Interrupt anytime. This is what we call the overall survival, and this is a survival difference. So meaning the other one showed you how long it took for the tumors to grow. This is telling you how long patients are living. And there's a big difference in survival already, which is good. When the data hasn't even matured yet, it's pretty good to find that. Here are the side effects. Not too much to speak about. Now, when we give this treatment, we are worried about the radiation being excreted through the urinary system. So there is some exposure to the kidneys. That's why we limit the treatment to 4 treatments. But we didn't see a lot of side effects to the kidneys on this treatment. In our clinical practice, we haven't seen very much. So the 4 treatments are very safe. Other toxicities can be bone marrow, meaning sometimes the lutetium dotatate can affect the bone marrow and decrease your blood counts. But in this study, it didn't affect anyone's ability to finish the treatments. Anyone have questions about that? Yeah.
Did anyone in that respect have to postpone one of the phases of their treatment, of the 4 treatments, because of blood count?
So from the graph we saw, I think it was 80% of the patients received all 4 treatments. I think delays are okay if they were to happen. And our experience, Sandra, do we have a dose delay for bone marrow toxicity? I don't remember one. Usually we had dose delays for other things like logistical issues, or someone had another problem, medical problem, and they couldn't come in because a lot of our patients came in from far away.
But not because their blood count wasn't
I don't
correct.
think we saw too many of that.
We
No.
didn't have any, I mean, the most we had like low platelets, but nothing to be clinically.
Right. And then the 8 weeks is plenty of time from our experience to recover. Now we do have patients who have received more than 4 treatments. And then we start to get into the issues we've discussed. So if you were to, the FDA label is going to say 4 treatments. So if you were and your doctor got together and decided that you needed more than 4 treatments, I think that's when you would start to get into this area of side effects, I would say. Probably a little bit more.
This is intravenous treatment, yes.
I'm sorry?
Intravenous treatment?
Yes. Okay, this is looking at kidney function, because everybody is worried about kidney function. And if you look, it looks like the kidney function did fine. It didn't really go down, which is great. Okay, so summary and conclusions. And I'll give you my perspective. This is really, it's great. This was a great study. It took a treatment that we already knew was working in centers, specialized centers around the world, and through a collaboration between AAA, investigators, scientists, patients, we all came together. Some of the patients signed up for a randomized study knowing that they weren't going to get the lutetium, but they did it. People worked hard. Sandra did a lot of work on this study. It wasn't easy. She's still doing a lot of work, answering queries every day. And everybody came together, and we actually did it. I think it's going to be approved by the FDA, and I think this is it's a great new thing for the patients, and I think it kind of establishes neuroendocrine tumors as a type of illness that we can really beat, that we can do something about it, and we can do some serious things about it. And I think that this will be a platform, and you'll see, and I don't know if Ayala her company's already working on it, but getting new treatments, new ideas. This is a new kind of approach to this disease, almost like a new approach to cancer, I would say. When you take an antibody to a hormone and you combine it with radiation and you show benefits like this, it's pretty unprecedented. So I think this is not an end, but I think a new beginning is the way I would look at it. Any questions about that?
What, is it being done the same way it's being done in Europe, where patients stay overnight or 2 nights? I don't
So
know
that's how it was. That's how the study was done. So you came in, you had the treatment, you stayed overnight for amino acid which would take the 24 hours also, and you'd stay overnight.
So like an observation, you know.
I think that it's going to depend. Dr. Lurie, maybe you can help us with this. Is every institution going to keep patients for 24 hours because of radioactivity, or do you think that?
So this study was from Midgut.
Right.
So what if you have a PNET or?
That's a great question. What do you think?
I'm gonna hope you're gonna say yeah.
So I think that's exactly what everyone. I feel like I'm at an investigators meeting because that's exactly what comes up, right? So I think that we're hoping that what we've been able to understand from this is that the biology is important, meaning the somatostatin receptor expression. So if the FDA agrees with what I think the investigators believe, then they will approve it based on the somatostatin receptor expression and not focus so much on the site of origin. Because as someone already mentioned today, sometimes you don't even know where it's from. That would be even a harder situation, right? So I think that we're hoping that the FDA will allow that. And we're hopeful, we're hopefully optimistic. If not, then the next study is going to be somatostatin receptor tumors non from midgut, and that'll be the next step. We'll call it NETTER-2, and we'll go from there.
Well, if not, can you use off-labels?
That would depend on the insurance company. I think the doctor would feel comfortable, and it's off-label, but there is precedent. There's lots of studies showing that in non-midgut tumors that are expressing somatostatin receptor, it's a safe and effective treatment. So that would be outside, no one can predict what Anthem's going to say, for example, or Blue Cross, we don't know what they would say.
Were people with tricuspid valve involvement included in this?
Well, that's a good question.
Excluded?
What do you mean, right, involvement?
Well, if you have right side heart failure, would you still be a candidate?
Right, so the inclusion criteria for all studies exclude patients with heart failure that's uncontrolled. And if you have controlled heart symptoms, then you should be okay. What I would say is if you have really bad tricuspid problems, we should get it fixed, and then you should go on treatment. Or if it doesn't need to be fixed, then you should be able to safe to go on treatment. So that's how I would figure it out. But you're right, that issue did come up. We had patients who presented asking for this treatment, but at the same time had pretty significant tricuspid valve regurgitation. We tended to repair it first, and then afterwards proceed. Yes?
I have a question. You had mentioned that the nucleotide scans were good because it helps the patient and the doctor know if they're going to be receptive to certain drugs.
Exactly, yes.
And what if at the point of surgery and biopsy, that test was not done? Is it something that can be done with the tissue after it's been removed from them?
That's a good question. Well, no, the way the test is administered, it has to be in your body. There is a somatostatin receptor that you can check in the tumor. I think they do it at Iowa.
So the slides at UCLA like would have mine and those.
You could send it, you can maybe even ask your doctor at UCLA if their lab can look at that. There are studies looking at serotonin receptor expression in tumor tissue. And there are laboratories around the world that do it. It's not a commercially available test. What I would say is that if you don't have any tumors, that's great, you don't need this treatment.
Right.
Hallelujah. And if anything were ever to come back, that'd be the time.
To do it to that too.
Yeah, I think that'd be a better focus than trying to figure out what in the past, because you don't even know, whatever the past results show, even if it did show you had receptors in the tumor tissue, when it came back, you would still need to do it again because you would hate to make an assumption on the new tumor.
With the patients for whom this was not effective, was there any commonality between them that anybody's aware of why it didn't work?
I have not seen any information like that. That's a good question. That's a very good question.
And
I think the best prediction for this medication would be the receptor expression based on the Krenning scale. That seems to be the predictor for treatment response. So that would be what I would focus on. So,
in other words, the more receptors present, the more effective.
Right.
You're saying? Now, is there any chance that after the 4 treatments, some metastases are still present and maybe they're hardened against the radiation and more aggressive after that.
No, I'm thinking that through.
I mean, you get rid of all the soft
So-
ones, but the hard
so-
ones.
Right, so, no, that's a good point. Okay, so radiation is a little bit different than chemo. So the way that radiation works is that it's pretty toxic. And in the way that it's delivered in this situation, I would say that any tumor cells that weren't killed, it's probably not likely due to resistance to radiation. It's more of a lack of delivery of the radiation. I mean, there are not that many tumors that are resistant to radiotherapy. There are, but if you give enough, it's going to do the trick. And I don't know if radiation necessarily can select clones that are resistant. I think that's what you're thinking about, kind of like a rapidly evolving Darwinian kind of thing where you kill certain tumor cells, then the ones you leave behind are more aggressive. We don't really see that too much in cancer therapies in general and specifically in radiotherapy. We don't really see that too often. So I would say that if it's not working, if there are tumor cells left over, it's probably more of a delivery issue. It could be that your tumor proliferative index is higher than you would want. And it's going to be hard for really small tumors that you can't see to receive this treatment, as well as bigger tumors that have a lot of receptors on them, I would say. But again, I'm kind of just talking. I mean, what I'm saying is based on what I think, not what I know. So from what we know, the only real prediction of efficacy is the somatostatin receptor expression.
I have one question for you and one for Ayala. I wonder if you could talk about PRT in context of when it's recommended. We've really done a deep dive into the study, but at what point, I know in Europe, it tends to be more of a first-line treatment, and the U.S. is maybe because it hasn't been available here later on. So when are you recommending PRT?
That's a great question. Wow, that's a great question. I think that's what everybody's asking from around the world right now. In all the meetings when we get together, neuroendocrine tumor doctors get together, we all sit around, we say, and the number one question is, okay, now that PRRT's been approved and everything's going to change, and then we're like, well, wait a minute. PRRT, I think, is different than other treatments in that it's going to be approved for 4 treatments, right? You can do more, but that's a limited amount of treatment time. So whether or not that's appropriate for somebody who's just diagnosed is questionable to me. There are other treatments that are very effective, specifically somatostatin receptor agonists. So you might want to stick with those. The other issue is if you have disease confined to the tumor or outside of the, if you have disease confined to the liver or outside of the liver, I mean, it gets complicated. From the way we approach the neuroendocrine tumors here is that we look at disease burden, we look at proliferative index, we look at whether the tumor is involved in the liver or not, if the tumor is in the bones, and how much symptoms do you have from the disease. It's kind of like those 5 things. And then we put them in our brains. We don't have like a written algorithm. We put that in our brain. And then we sit around the table, and we have a doctor, we have a surgeon, we have a medical oncologist, we have an interventional radiologist, and then we have a GI doctor, and we have a radiologist. And we sit around and we think about, okay, what are the tools that we have to treat neuroendocrine tumors? We have PRRT, we have, octreotide LAR, we have lanreotide, we have everolimus, we have surgery, we have liver-directed therapy, radioembolization, chemoembolization, ablations, and then we take them all together and we say, okay, for this particular patient at this particular time, which do we think is best? Most tumors, if you do those types of things, you don't really help. Most tumors, if you have stage 4 disease, you don't really operate too often. Most tumors, if you have tumors in the liver, you don't really want to do embolizations. You don't want to do radiofrequency ablations. But for these tumors, those treatments seem to work really well. So we don't want to discount them and say that none of those things are going to happen. What we have to really think hard on what to do is how to put PRRT into the treatment decision-making process. Where are we going to fit? I think with some more time, it's going to become more evident. I think here, our first line treatment for patients is going to be, if they're asymptomatic, if their somatostatin receptor expression is going to be SSRA still. If somebody has one liver tumor, we're probably going to remove it. If they have two, we're probably going to remove it, right? If there's maybe 3, 4, 5, if they've already had liver, you know what I mean? So I think we're not going to, now if you're the FDA, they're going to say, yeah, PRRT 1, and then, you know, that's it, right? Because probably SSRAs is not even FDA approved for a lot of the ways that we give it. So it's kind of, it depends on your, also the perspective you get from the regulatory versus the doctors. But it's a very important question. I don't think that for our patients day to day it's going to change very much. I still believe strongly that we're going to start with SSRAs because you can give those for several years at a time. And just like in the study, if you progress on that treatment, then PRRT will be recommended if you have extrahepatic disease. I think if you're symptomatic and you're not improved with serotonin, with the octreotide or lanreotide, and you don't think that liver-directed therapy is a good idea, they might make a shortcut. Or if you have somebody who presents, unfortunately, with lots of cancer, I think those would be special circumstances.
It just confirms how individual we all are. You know, each
Yeah, I
case
think
is
so.
unique.
And I think that's what's so fascinating about this type of cancer is that really, I would say in most other cancers, patients are not so variable and presentations are not so different. But in neuroendocrine tumors, there's quite a difference between person and person, and it's tough to put everybody in one box. It really is.
Thank you.
You're welcome.
Just a technical question. I know that there's different trials in the U.S. now, and for patients we're reading there's JR11, there's Y90, there's lutetium-177. Can you just speak a little bit about the differences of those and how they actually work on the tumors? And then maybe a comment on the amino acids that are used and how that differs from Europe these days.
Yeah, so indium and Y90, there's more radiation exposure. So it's not as limited and not as tightly controlled as lutetium. So you could think of it like this: if you have one particle, how much penetration there is, and the less penetration, probably the safer it is for surrounding tissues. So some of the radioisotopes, there's more of a broader effect. There's more radiation exposure. So that's a concern.
There was at the NorCal conference, there was a question about the size of the tumors and in Germany how they did a mixture depending on the size.
Right.
So it's like, is there a certain point over which you say, okay, this one's Y90, like is it 4 centimeters?
I think we should leave that to the Germans to figure it out because they have their own, you know what I mean? They have a very experiential approach to it. I mean, no one's done it more, and no one's combined the treatments as much as they have. So I think for now, especially, I think we're going to be limited to just lutetium because that's what's been approved. And I think it's best that way also because that would, God forbid, that we would start tinkering it around. I think once more understanding, I'm sure with this, there's going to be more research into this area. It's going to be, I'm sure there's going to be a lot of intense interest. And these questions, these are amazing questions. I think they're going to be answered. And then we can, once they're answered by the expert centers, then other people can, you know, probably take their lead.
Will the Houston Center continue their PRRT also?
What's that?
The Houston Center, are they continuing their PRRT?
I don't know. I hear different things. I'm
they're
not sure.
up and running now.
That's what I heard.
My other question was about the amino acids.
The European experience is that you don't necessarily need a cocktail. So it's, you know, the cocktail that we used for NETTER-1 did cause nausea, which I would say is #1 side effect of the study was from the amino acid cocktail, if you can believe it. But there are other cocktails that we think that are probably just as effective that cause much less nausea. And I think that if the drug is approved, we'll have more leeway as far as which cocktail we're able to use.
I was wondering if I might have missed it. Is there information about the size of the tumor of these patients?
I don't think that's going to be published, probably, the size. Is there something specific you're curious about?
Yeah, because I guess in response to Giovanna's questions about the...
Oh, to see if size of the tumor? Right. I mean, maybe the NETTER-1 investigators should look at that down the road, but I haven't seen any information. Yes, sir.
I'm trying to picture how this is working inside the body. And from what I can understand, the lutetium is spreading throughout the whole body, right?
Right.
And it's radiating the whole body. Maybe it clusters on these somatostatin receptors, and that's where it will hang around for a
No.
while. But how is it not affecting the whole body?
Well, right, so the way that it's engineered is that it's only released based on the binding of the antibody.
Is it in some kind of protective, I mean, it's a radioactive isotope, it's got to be shedding radiation constantly, right? What do you mean it's not released until it...
Well, it doesn't stay. It gets excreted to the urine rapidly. Yeah, I mean, it's in the vein, it goes through the system, but then it gets excreted out the kidneys. So the only radiation exposure that's really significant is the one that's to the tumor that's long lasting.
Okay, so when it binds to the receptor, it's going to stay there a while, but the rest is just moving in and out quickly. Delivering its radiation.
Theoretically, it's internalized. So when you bind to the antibody, those agents tend to get internalized. But at the cellular level, I mean, I'm not sure how much that matters. You want the radiation in that physical area. I mean, that's how radiation works, right? Now, the question about safety is important. You know, is it a safe therapy to use? Now, if the radiation exposure to the body was a problem, then the results aren't good. Patients don't do well, they become sick, they have side effects, they don't live as long. But in this one, all the treatments were positive. So you can do all the, fancy, theoretical models that you want. You can do basic science experiments in the lab. You can look at the tissue, but the bottom line is you see how people do. And if they do better, then, the theoretical risks are outweighed by the benefits.
At what point, or is there a point when one human body has its maximum dose of radiation? Is there a point at which you say, all right, no more?
Well, the FDA is going to be approved for 4 treatments. After that, it depends on the body. It depends on, I would say, how much tumor they have, how much exposure radiation they get. This treatment, if you give it to someone with no tumor, or you give it to someone with lots of tumors, the radiation exposure is quite different. And the dosimetry is not very straightforward. It's definitely beyond my ability to calculate. So, even a physicist would have a hard time really estimating how much radiation that you're getting. So I would say bottom line is when you give a treatment like this, you assess the kidney function, you assess the bone marrow function of the patient. Those are where you know that potentially there could be trouble down the road. And if they don't have problems in those areas, I think that you're probably safe to proceed. Now there does come some issues. You have a patient who was treated with Y90 in the liver, someone who had external beam radiation therapy. So let's say they had leukemia, a different illness in the past, and they had radiation. Those are different. Those are rare cases that do come up, and they require lots of thought and conversation and consideration. But it's not something that you can just quantify and just say this is the answer to your question. I think it's going to have to be case by case.
What's the difference in the binding to the receptors of like the lanreotide versus the PRRT?
It's the same drug. It's the same drug. But lanreotide works by decreasing the hormones that feed the tumor and spur its growth. This is actually utilizing the hormone to bind to it, radiation therapy. So it's a different kind of treatment. Yeah, but you're right. It exploits that binding to the lanreotide or octreotide, right? That's what makes it pretty neat. Okay.
Great.
Yeah.
Thank you so much.
No, you're welcome.